Our next panel consists of Nicole Willett, Robert Zubrin, Carol Stoker, Chris McKay, and Reut Abramovich. They're going to be speaking to us about the search for life on Mars. Welcome.
Hello, everybody.
Hello, Nicole.
Thank you for joining this all-important panel. And thank you, Dr. Zubrin. He was a surprise. He wanted to join. And thank you, Dr. McKay and Dr. Stoker. And it's so nice to meet you, Dr. Abramovich. So today, I want to ask questions sometimes to certain scientists, and sometimes you can all just chime in as you wish. And I would like to start with Dr. Stoker and Dr. McKay about questions I got from Dr. Gil Levin. He sent me some questions.
Great.
You both spoke of him yesterday, and I am one of his biggest fans. And he would like to know two things. Has phosphine been sought or found on Mars? And is there any known factor preventing current life on Mars? So those are his two questions. Have at it.
Carol, are you there?
Well, I'm trying to figure out where my video is.
You're on the moon, so I would try
Yeah, I can see the moon.
Tranquility.
See my face. Oh, well. We'll have to
While
go
we're
without.
waiting for Carol, can I just interject two comments, Nicole? One is the previous question about, from the last event panel, how to get nitrogen on Mars for biology. One would not extract it from the N2 in the atmosphere. We know there's nitrate in the soil, so you just dig it up. It's pretty thin, but it's much higher relative concentrations in the soil as nitrate. And plants and microbes will be happy with nitrate. The other thing I wanted to do was just say hi to Reut. It's great to see you, friend and colleague for many years, spaceward bound, and so on. But anyway, Carol, if you wanted to address the question Nicole raised, that I will add my comments too.
Yeah, so what you're going to see is the moon, and you're not going to see my face because for some reason the background is there, but the person is not. I don't know why. It's changed. It used to work. So the question was, better remind me.
Has
phosphine been seen?
Been sought?
To my knowledge, no. In fact, phosphine has suddenly been seen on Venus, and is now a big buzz that Venus is habitable because they've seen phosphine on Venus. I know of nothing that would have, no report of phosphine on Mars.
Okay, thank you. And Dr. Levin also would like to know, is there any known factor preventing current life on Mars?
On the surface, yes, there's a few items preventing life on Mars, but on the subsurface and perhaps underground lakes, there are many places where life can be sustained and can be very happy on Mars.
Wonderful. And since we brought up Venus, I'll ask Dr. Abramovich the Venus question. I would like to know, what are your thoughts on the buzz around life on Venus, and what type of life could it be if there was life on Venus?
So Venus doesn't have really a good habitable area above surface. Closer to the ground, you have really high atmospheric pressure, and you have high temperature, which we know now from hydrothermal vents. It's actually a place where life can be alive. There's a lot of nutrients going around in Venus that goes for it, but the issue is a lot of the sulfuric acid, which is in the atmosphere, and that's also apparent in the upper atmosphere, and that's pretty deadly. Like the pH levels are very, very acidic, and the water availability is very, very low in Venus, so we think it's not a good place at the moment for life. But having said that, I would love to have a Venera D mission going to Venus again, and putting our technologies up front and seeing what's happening there. Today, we can do a much better mission at exploring Venus on the ground and also in the atmosphere. So as an explorer, I would like to go and check it out for sure.
Yeah, there's been some talk about maybe some life in the clouds on Venus. So It'd be interesting to see if that happens. And
It is.
sorry, go ahead.
It's okay. Go ahead.
Dr. McKay, yesterday in your morning plenary, you spoke of a non-life detection mission to Mars, and also whether there was a second Genesis on Mars. Can you explain how a non-life detection mission may miss out on identifying a second Genesis that utilizes a different genetic material than what we would be looking for?
Okay.
And how that may affect a human mission to Mars?
Sure, that's very good question, very good point, and the 2 points do go together. The point about a non-life detection mission is really a check the box exercise, if you will. We want to put humans down in the equatorial region of Mars, in an area which most people, look, Carol's got a good picture of it as her background there. Most people think that these areas of Mars are lifeless. Gil Levin's results notwithstanding. We think that they are highly oxidized, very low water activity, even lower water activity than the water activity in the lifeless clouds of Venus, and exposed to high UV and even high ionizing radiation. But we don't know for sure that it's lifeless. One reason we don't know for sure is Gil's results, for example, can be explained by biology. That's not most people's preferred explanation, but I think it would be premature to be dogmatic and say, we know there's no life on Mars. It's safe to bring humans back. So I think there's going to be a requirement to either quarantine the astronauts from the Mars dirt or on their return to Earth, or conduct a due diligence experiment on the site similar to where they're going to confirm to some level of reasonableness determined by scientific community that there is no life. Unfortunately, the Viking results don't meet that standard. None of the subsequent missions meet that standard. So we need a mission, in a sense, to show that there's no life in the equator of Mars, right? If such a mission was a hitchhiker along, say, a SpaceX large lander, the total cost of it would be probably $100 million, a drop in the bucket compared to any incremental cost on human exploration. So that's what I mean by a no life detection. And as Zubrin so nicely pointed out yesterday, that would not prove there's no life on Mars. Of course not. It would just prove that locally and temporally there is no life within some reasonable detection limit, making it safe in a sense for astronauts. But there could very well be life, as Reut just indicated, deep below the surface in an aquifer, happily metabolizing along like we know is occurring on Earth today. And that life would not contaminate astronauts. It would not impair their ability to return to Earth, but they could discover it through deep drilling. And then they could maybe discover that it's very different from life on Earth. And that would be very wonderful. I hope to live to see that day. Well, even if I don't live to see it, I can imagine it, because that would tell us that there's 2 life forms in our solar system. N is greater than or equal to 2, where N is the number of life forms in the universe. And that is really quite a different question than the biohazard. How would we, your second part of your question, Nicole, is how would we know that it's a second Genesis? And I think the operational answer is we would see if it maps on the tree of life. All life on Earth maps onto a tree, a phylogenetic tree showing the relationship of that life. Viruses sort of float around the tree, but they are on it. They're in the tree, maybe in the bushes or the roots or somewhere, but they're in there too. If we find life on Mars and it doesn't relate to that tree at all, even if it's made of carbon and water, and even if it's made of the amino acids, if it doesn't relate to that tree, it's a second Genesis, N equals 2. And we have achieved an amazing scientific step forward in our understanding of life in the universe. And that would be wonderful.
It would be great. And I have a question for you, Dr. Zubrin, since you brought up some of the stuff, Dr. McKay, about what Robert Zubrin pointed out yesterday. Can you talk about the planetary protection? I know you have some strong feelings about that and life on Mars, and how NASA and other space agencies are, you know, so worried about the planetary protection. Can you delve into that for me?
Well, sure. But see, I have a somewhat different point of view than Chris on this issue, because he wants to send people where there is no life, and I would want to send people where there is life. Okay, that is, I believe that the purpose of sending astronauts to Mars is not to send them to the least interesting places, but to send them to the most interesting places. And certainly if we sent a life detection experiment to Mars, I would want to send it to the place where I had the best chance of finding life. And as Reut said, on the surface, it's a pretty outside chance, but you might have a chance if you sent it to the edge of a glacier and you could drill down very easily through a glacier, and maybe there's a liquid layer at its base. That has a chance of being a biologically active area within easy reach of an atmosphere. Of course, if we could drill down 1 kilometer and get to the groundwater, that's the best chance. But on a first human mission to Mars, your best chance of finding life would be, in my view, under the ice rather than under. So at mid latitudes, 38 north, there's glaciers. That's as far south as San Francisco is on Earth, about where Chris is sitting right now. So that's a dangerous area, Chris. There are microbes there. But the yeah. But to be clear, look, the fact that there's life somewhere doesn't mean it's a pathogen, okay? And it's really unlikely we're going to find bat viruses on Mars, okay? Because there's no bats there, and you know, or any related megafauna or megaflora for that matter. And so the idea that we'll find pathogens that are adapted to attacking animals like us it makes no sense. And furthermore, as Chris said, you can't prove a negative. If you landed a lander and you picked up a sample of dirt and you examined it in various ways, it wouldn't prove that there wasn't microbes in the dirt 10 meters away. So to anyone who actually thought this was an issue, you would not prove anything. So I'm all for sending life detection experiments to Mars. And if human missions are delayed, this to me would be as best as we can do, send robots to find life. And if we can find life or anything looks like life, then definitely that's where I want to send the people. The idea that any dirt from Mars represents a biohazard. We have rocks from Mars landing on Earth all the time. Trillions of kilograms of rocks from Mars have landed on Earth since, 3.5 billion years ago, when there was liquid water on Mars. And, one thing we found from the Allen Hills meteorite examinations, Professor Kirschvink found this, and through his examinations, was that major parts of that rock during its entire career of ejection from Mars, cruise through space, and re-entry and landing on Earth had never been raised above 40 centigrade. So that if there had been microbes in those rocks, they would have survived the trip. And if the Red Death could come from Mars, it's already here. And this idea that we should sabotage the science of human exploration out of fear of going to interesting places, just as the Curiosity mission has already been degraded by not letting it go to places that it looked like there was water seeps for fear of planetary protection issues. And then, so that's the back contamination, and the idea of quarantining the Mars sample return and even destroying the sample if they found that containment had been violated is so crazy. It's like, the customs officers looking in the trunks of everyone's cars coming into the country from Canada, searching for Canada geese, when meantime they're flying across the border by the millions on their own. And then there is one other issue, though, that I've heard raised by astrobiologists, some of them, at various conferences that I've attended in the recent period, who were not so concerned with this back contamination issue, but with the forward contamination. That is, their argument was, we don't want to send people to Mars, because if you send people to Mars, you're sending microbes to Mars. And then how would you know that if you found microbes on Mars that they were there before you were there?
Right.
Well, how do we know there was life on Earth before we got here? We know because there's a fossil record, okay? If there was life on Mars before people got there, they will have left fossils and other biomarkers.
Mm-hmm.
So if you find life on Mars, which is different in any fundamental way than life on Earth, for instance, it doesn't use DNA, it uses some other information system, well, then you know it was indigenous. If you find a life form that does use DNA, but it is unfamiliar with any kind we've ever seen on Earth, then it's also indigenous. If you find a life form on Mars, which is identical to familiar bacteria on Earth, but you also find biomarkers, then it's indigenous. But if you find life forms on Mars that are identical to Earth life and no biomarkers, then yeah, you brought it. So we can easily tell what if we find life on Mars, whether it was indigenous or not. And to think that there could be biomarkers on Mars and not be indigenous life, it's like creationism, who say, you know, how do we know that there was ever life in the prehistoric past, because God could have created the earth with the fossils in it in order to test our faith. So, you know, I don't see any merit in the planetary protection arguments in either the forward or back contamination cases.
So
And
there is
rather-
a spoken like a non-biologist.
Exactly.
Non-biologist.
But that's okay.
Well, yeah, but
Spoken
of
like
course,
somebody who
most
doesn't
of
know
us
what
are
he's
really
talking
gonna
about
hear rant.
and is monopolizing the conversation, spewing out dogma instead of focusing on the issues. This panel, Bob, is the search for life. It's not
Yeah?
a rant about your particular issues with planetary
hey,
protection
excuse
policy.
me, I was asked the
Forward
question.
protection's got nothing to do here. You're just a guest on the panel. Pipe down and listen.
No, okay, stop it, Chris. I'm
I
being very
think-
polite to you.
Okay.
Thank you,
That's
Nicole.
good
Take
for me.
charge. Thank you.
I think Dr. Abramovich had something she'd like to say in response to Dr. Zubrin, and I was actually going to go to you next anyway. So go
Thank
ahead.
you very much. Thank you. It's a fascinating topic, and part of my recent work is actually addressing part of this issue. So one of the things I've done so I'm an astrobiologist. I work with stromatolites in Shark Bay and extremophiles, and recently I'm into cross-contamination issues, which is exactly the point here. And one of the things I've checked in the last a year or so in an analog mission here in Israel is exactly the issue of how you monitor a contamination situation on Mars. So you have a crew of astronauts going on Mars, and they're coming with their own bacteria. Their equipment's going to have a few spores on it. Everything is fine. So one of the methodologies that I'm looking into is exactly checking out from a spatial and temporal point of view how much contamination is going into the ground from the astronauts and their equipment, but also backwards contamination, looking at the extreme environments and coming back in. So I totally think that we should go to Mars. I totally think we should check for life there. There are many different technologies today to check out if it's indigenous life, if it's not, what type of shape. But also you have to understand that from an astrobiologist point of view, 4.5 billion years is a very long time for a lot of evolution to happen, and a parallel evolution. So even if we find something that is different from what we like, we can still, I think we can detect it today. We are quite aware of all the different biological pathways there are there. We're not 100%, we don't know everything, but we have the technologies to go and check them out. So if you go and you monitor what's happening with the surface, with the subsurface, and you do this on a spatial grid in a temporal fashion, you can check the dynamics of what's happening because of the astronaut crew, but also how the environment would affect it back. Now, sending samples back to Earth, I was always an advocate of it's okay to bring samples, but not to Earth, but perhaps for the International Space Station or any sign of an outer space signpost designed as a biohazard level 4 lab or something like that. And now we have the capabilities of flying PhD students and scientists to the ISS to do the work if needed. So that would be some sort of a more secure fashion of than actually bringing the samples back to planet Earth if we're, you know, afraid from that point of view.
So I would like to bring up what I think is completely missing in Robert's analysis, and that is he's thinking of it only in terms of pathogens. We're going to get the Andromeda effect, you know, and it's to me the much bigger risk is not pathogens. It's alien invaders. It's invasions of things, microbes most likely, that are sufficiently evolved or different, whether they came from Earth or Mars and Earth shared a common origin or not, but they are sufficiently changed that when brought back to Earth and getting established on Earth, they might upset the microbial balance of the biosphere. And the microbial balance of the biosphere is actually what controls the air we breathe and the conditions that we're living on. If you start messing with that, it could be very catastrophic, much more catastrophic than a disease. And until we actually know that that is not a risk, we should not be bringing stuff back from another planet. Full stop.
Let me amplify what Carol said with an example. Rabbits in Australia are not pathogens. They're not dangerous. They're cute and cuddly. You know, some of my best friends are rabbits in Australia. So it's not the problem that the individual organisms are going to attack us. As Carol said, what they've done is upset the ecology. Now
we have lots of examples of that on Earth.
Yeah.
Lots and lots.
And I think the point is not that we cannot bring a sample back, but we have to bring it back in a way that's contained and in which it can be carefully and safely transferred to a containment facility, as Reut was saying, right? We know how to we know how to deal with dangerous organisms. We do it on Earth all the time. But if the astronauts are coming back with dirt in their boots and dirt in their lungs and dirt in their hair, that's not an easy thing to contain. And so my point here is that we have to resolve the question of sample return to Earth before astronauts are coming back to Earth. Now we can resolve it by bringing a sample back robotically and searching through it carefully and so on, or we can resolve it in situ on Mars at much lower cost with a non-life detection mission. But the point is we need to resolve that question before astronauts are coming back, or the knock-on costs and effects to the astronaut program will be enormous. Think of the costs associated with the quarantine of the Apollo astronauts. And now amplify that by orders of magnitude, because it's coming from a planet where there's a real risk, not just an imaginary go out and shake the president's hand and then get back into a container, where there's a real risk. And so I think that from a purely cost-benefit point of view, the human exploration program needs to take this question seriously in terms of when the phasing is between humans going to Mars and sample return. Now, it wasn't that many years ago when the phasing was simple. Neither sample return nor humans were going to ever happen, so we didn't need to worry about it. Now what's happening is there seems to be a sample return program almost going in at NASA, and human exploration is racing ahead. All good news, but no one's thought through how does this play together in dealing with the very real issue of sample return.
So,
Okay, well, let me just say in answer to that, just briefly, as far as microbial invaders attacking our microbes are concerned, you know, once again, SNC meteors have been coming to Earth for billions of years, and somehow the Earth's microbes have taken care of themselves. But in answer to what Chris just said, I actually agree with you. I agree with your course of action, not with your motivation. That is, I actually think that a much more effective robotic Mars exploration program would send a large number of rovers to Mars, to a large number of locations with all sorts of different suites of instruments, and do all sorts of studies in situ. And I think that's going to tell you more about Mars, including the possibility of finding or not finding life on Mars, than a sample return would. So I'm actually there as far as that's concerned. That is, I'm not that excited about sample return. If we're thinking in terms of a fixed or reasonably fixed budget of a robotic Mars exploration program, I think we'd have a much richer program with large numbers of rovers to large number of places with lots of different instruments looking for all kinds of things. I don't think, I mean, look, if you're going to do sample return, the whole argument for sample return is you're bringing the sample to Earth and where it can be looked at in 1,000 different university laboratories with all sorts of instruments that are not available in a limited facility, such as the space station. So you're going to do sample return, you want to bring the sample to Earth. But if you're not prepared to bring the sample to Earth, you shouldn't be doing sample return. You should be doing in situ science in a much richer and more diverse way. So as far as that part is concerned, I'm with you. I'm all for life detection experiments, but I don't think life detection experiments are useful in trying to prove that there isn't life on Mars, because if you're going with this what if scenario, which is really what you're doing, you're going with very low probability risks, but you're saying if there's a finite possibility and it could have a catastrophic effect, you know, reducing it from 1 in a million to 1 in 2 million chance doesn't really change the math very much here. I think we should be sending life detection experiments to Mars for the purpose of detecting life and then following that up with people to study that life.
Okay, so thank you. I would like to follow up on something Dr. McKay said about bringing back a sample return and containing it. You know, in 1996, Dr. David McKay came out with his team and said, we found this bacterial fossil in ALH840001, the meteorite. And it's still being debated to this day, back and forth, back and forth, back and forth. So if that is the case, and we bring a sample return from the cache from Perseverance in a few years, whenever that will be, and then we study it for 20 or 30 years, and argue back and forth, how is that going to affect a human mission to Mars?
Well, you know, you've raised some very important points, Nicole. First, on the meteorites, ALH840001 and all the other meteorites we have from Mars are volcanic rocks. It appears to be that those are the only rocks that can survive the ejection into escape velocities. They're not very good samples for a search for life, so it's not really a good approach. And the controversy behind David McKay, who, by the way, I'm related to through a least common universal ancestor, 3.5 billion years ago. What they found was interesting evidence of water activity, and they sort of stretched it a bit, and they got in trouble, I think, on that. But the rock is very interesting, and it is a window into early Mars that was wetter than the current Mars, that's for sure. And the meteorites in general, like that. Bringing a sample back, I think, you know, if I was in charge of the sample return program, the first sample I would bring back would just be a grab sample of the loose dust, the same kind of loose dust that Viking sampled, the same kind of loose dust that the astronauts are going to breathe, the same kind of loose dust that is inevitably going to get carried back with astronauts. And I would bring that sample back because that's going to be important to the engineering design of the human habitat. And it will also be in a very important achievement of due diligence in life detection. We look at that dust and say, hey, there's nothing alive in it. It's safe for the astronauts to breathe this. It's not going to make them sick. And it's safe for them to bring bits of it back home, inevitably, which they will, right? So the first sample return in the program that I would outline would be a grab sample of that. Now Perseverance is not going to do that. I don't think they even bring any of the loose dust back. They just bring rock cores back because it's a mission driven by hard rock geology. So I'm not sure it's going to really give us much insight into life on Mars now or ever. I think it'll give us a lot of insight into petrology of the volcanic rocks on Mars. And that'll be good for a paper or two in JGR or meteoritics and planetary science, maybe even Nature Science. But I don't think it's going to give us much about biology. Now, I may be wrong, and maybe the instruments on the rover itself will find very interesting traces of carbon and biomarkers that would, make us really want to have that sample back. But I don't think so. I think that we're going to find that they're rocks. They have mineralogy like the rocks that we've been studying on missions to date. And the geologists will love to have it back, but the astrobiologists will not. And it may actually undercut the motivation to spend the money to bring it back. We may analyze those rock cores and decide they're not worth bringing here. Just leave them there on Mars.
Yeah, so actually Adrian Brown gave a presentation yesterday about they're going to search for stromatolites, right? Well, that's a very optimistic scenario that they're going to find stromatolites. But what I think they could find is evaporite deposits. And that is a sample that I think we should be pushing for. That if they find evaporites, in particular halites, that they bring that back. Because that's probably the only place that you got a chance of having a modern biological signature in the place that they're going.
Is the core capable of sampling halites if they came across a salt flat?
Yes, I think it is.
Okay.
So I was going to ask Dr. Stoker, yesterday during your plenary, you showed the slide of the climate model of the thermodynamically possible pure liquid water on the surface of Mars. I know that was a mouthful, but can you explain, and maybe Dr. Abramovich can come in on this as well, how is that important to the potential for finding extant life on Mars? And could that help in that search, knowing where those areas are?
So I think what you're getting at is that in order to look for a biological signature that you're going to have any hope of finding, you have to go to an environment that's habitable. That's actually been recognized for quite some time. It's just that where are those habitable environments is slow to get acceptance. I think we have finally started to convince the community that the circumpolar icy deposits on Mars, where we're trying to send the icebreaker mission, are habitable environments, or at least sometimes they are. They're not currently habitable environments, but they have been habitable in the last 5 million years. So the ice is certainly one of the attractive places that you would want to go to look for life. And the ice is by itself and at its current state, quite possibly too cold for biological activity. So it might have periods where biological activity can occur as climate goes through these cycles, which are actually pretty high frequency cycles. So, time scales of 100,000 years, you get really radical climate change on Mars. So I think the icy deposits are a very high priority target. I also think that the salts are an interesting and another plausible target. Because these kinds of salts and salts that occur on Mars, like perchlorate, are very hygroscopic, and they will pull water out of the atmosphere when the atmospheric humidity gets to something like 75%. So that is less than is required for frost to form on the surface. And we do see frost forming on the surface. But those hygroscopic salts will pull the water vapor into the salt crystal and actually fluidize the salt crystal itself. And you can watch this under a microscope. You can watch a salt crystal suddenly turn into a blob of water.
Right,
and
like on the Phoenix,
Mars-like conditions.
like on the Phoenix, the struts of the legs of the Phoenix lander. The salt was pulling the water into the globules on the legs.
Well, yeah, well,
something to
Phoenix
that.
lander leg was actually, I think, highly overinterpreted by the person who wrote that paper, because basically you had jets, the landing jets themselves, which were at something like 1,000 degrees, were heating that water ice and causing it to condense on the lander legs So, you know, that was a highly non-Mars-like environment for the time scale that those drops formed.
Right.
And were those drops or not, I think is also speculative, because the camera that you were taking the pictures with was super low resolution, and you couldn't tell if that was a crystal or a blob.
Okay. All right. So I was going to
Those images.
Oh, thank you. I was going to follow up with Dr. Abramovich. What kind of extreme organisms could live in a very cold, very low humidity, salty area on Mars or on any other planetary body for that matter?
So we usually find extremophiles in different areas on planet Earth that are very capable of living in the conditions you just mentioned. And each one has their own mechanism, starting from the physical niche where they reside, for instance, and then goes to how their membrane is formed, what kind of enzymes they're working with. And you can see these modifications even in their own genetic code. It's really hard to say what kind and how they look. Most probably they're going to be microorganisms because they are best adapt. Some microorganisms have like this biofilm, like a gooey stuff around them that gives them their own, you know, humidity and nice area to develop. Caves are especially a big wonder because some of the microorganisms there actually evolve into introducing minerals. They look like minerals. They introduce minerals into their organelles, internal organelles. And we really have a spectacular diversity of life here on Earth. And we have discovered a lot in the last decades using molecular techniques and very interesting techniques. What I'm very much interested to see is a mission, whether to the moon or to Mars, which will include more molecular tools, more microbiology attitude. I have to tell you that when I go to a field expedition, there's a big difference between geologists and how microbiologists sample the area. So today, it's very clear that the people behind the amazing science that has been done on Mars are very much from a geology background in that area. But there are so many things today that we have learned how, as a microbiologist, you need to approach an area, how to sample it, how to not disturb the life as it is in its own environment, and make sure that the sample you're taking is actually a good representation of the area that you're looking in. There's so many different things you have to pay attention to, which geologists are not, as they have their own issues to look at. From a marine biology perspective, we have a lot of new technologies today that can go very deep, very into really high pressure areas, that can explore autonomously big areas, whether it's underground caves or, you know, in oceanography, we have gliders. We have sort of like an in situ microscopes today in marine biology. They're called cytosubs. And basically what happens is that this really nice thing, it stays underwater. It can attract the water inside, and it can beam a laser onto any particle that's in the water stream. And then you can get a lot of data out of that wavelengths that come out. You can know if there's chlorophyll, if there's something else. It takes pictures while it's doing it. So all these things could be used perhaps in a mission in which you take some samples from the dust, as Chris said before, and all these things, and you can sort of like dilute them with a bit of water or a bit of liquid, and you can check out, and all these data can be transmitted back to planet Earth. Having said that, all these things, and NASA Ames have done a spectacular work in looking for equipment that can do several different types of tests and find these life signature. I just want to point out that we do have life signature today on the moon that could be a good exercise ground for that. And that's for your consideration.
Awesome. I was going to ask about the NASA scientists. I don't know about the Israeli Space Organization, if they would have any plans for this. But I was going to ask if NASA has any plans to send a DNA sequencer to Mars, because a few years ago, J. Craig Ventnor proposed that, and there was a bunch of articles about it, and I've reached out to his organization several times to no avail. Do you think that would be a worthy mission, to send a small DNA sequencer, since they're getting smaller and easier, and pretty much anybody can send away for their DNA for $50 these days? What do you guys think about that?
Nicole, that's a very good question, and it's a very good idea. And I would just change your question a little bit.
Okay.
Can we send a sequencer to Mars that could sequence, among other things, DNA? And the answer is yes, and we should. There's recently been invented so-called nanopore sequencers, in which the molecule of interest is drawn through a little tiny pore, and it's sequenced that way. And it can be used to sequence DNA and RNA. And there's a company, I probably shouldn't name the companies because that would give NASA endorsement, but there's companies that are making commercial units that are the size of thumb drives, which can be used to sequence DNA with this nanopore technology. But the important point is that this would sequence any linear molecule, right? So a general argument has been made by Steve Benner, a very brilliant biochemist, that life must have a linear molecule to store information. It doesn't necessarily have to be DNA, but it has to be a linear polyanionic molecule. It's a very elegant argument. And for those of you who don't know much about it, you can read up about it. It's worth a study. So even if life on Mars does not use DNA, let's hope it doesn't, that would be very interesting, but it uses some other different kind of linear polyanionic molecule, as Benner is suggesting is required for life to have storage capability, information storage. This nanopore sequencer would still sequence them. So we're not proposing to do PCR, for example, which is specific to DNA, or to do any kind of sequencing that's specific to DNA. I think that I'd be disappointed if we found DNA on Mars. I want to find something interesting and different. So we've been focusing on these nanopore sequencers and trying to develop ones that will survive space flight. The challenge is making them rugged enough that they can survive a long trip to Mars and operate under Martian conditions, et cetera, and not being babied in the lab. And we've made great progress. So now your specific question is, are we planning to do that right now? There's no mission planned. Even our icebreaker mission doesn't include one of these, although we discussed it. It's not, the technology's not quite ready for flight, but it's getting there. And we will soon, I think, include these sorts of things in our missions.
Thank you. And speaking of Dr. Benner, I have a question here from Bob Bruner, who I think sent his questions out to everybody a couple weeks ago. He was asking about Steve Benner's theory of life starting on Mars, because Mars had the boron to stabilize the ribose and RNA versus the Earth, which didn't have the boron, and panspermia, and maybe we came from Mars. I think several people have now proposed similar theories. That's open to anybody. And if you want to argue, more ratings. So go ahead.
Well, there's no evidence, plus or minus, that the life that is on Earth did originate on Earth. Okay, it might have, but there's some questions about that, going back to Arrhenius, in that the simplest free-living organisms we find on Earth are bacteria, which are actually extremely complex things. And so, he brought up the idea that it could be an immigrant phenomena. Now, his panspermia is discredited because he believed that the universe had always existed and therefore life had always existed. And so the origin of life for him was not a meaningful question. But if we don't adopt that idea, but simply say, let's open up the constraints on what the conditions were where life originated. Okay, because if it did originate somewhere else, there could have been different conditions. There was an experiment done in the 50s, the Urey Miller experiment, where they synthesized a whole bunch of amino acids. But people said, well, this is not relevant because the conditions you had in your experiment were not those of the early Earth. Well, perhaps they were those of the place where life did originate. So it's very interesting to go to Mars because, okay, if we find a different kind of life, doesn't use DNA, uses a different linear sequence, that would suggest independent origins, well, clearly independent origins, but that would suggest that life on Earth could be an indigenous phenomenon because we have two separate examples of independent genesises. If we find identical life on Earth and Mars and no prior evolutionary organisms. That could suggest they're both seeded from spores from interstellar space. If we find life on Mars that is of the same general type as Earth life, but also including more primitive organisms than bacteria, then that would say that life on Earth came from Mars. So there's a number of possibilities here, and the only way to resolve it is to go and look.
This is really quite remarkable. I would have given exactly the same answer as Robert Zubrin. I think this is the first time we have ever been in agreement. The only difference is I would have been shorter in my response.
Thank you. That's good to know. We're moving on to the same page. Bob Bruner also wanted to know about how you guys feel about the Murchison meteorite and several other meteorites now that have been found containing amino acids and other organics. And in my opinion, I feel like the building blocks of life are everywhere we look. We find them in comets, meteorites, everywhere. There's enough chemicals, minerals, molecules on Mars to have life survive there under certain conditions. So what do you guys think about, could life be everywhere? Do you think life is everywhere? What are the odds of us finding life on Mars or going to Proxima b and finding life there based on the amino acids and organics we're finding everywhere we look?
Well, I think, Nicole, that is one of the reasons why we may be optimistic to find life, why people like me and Carol and Reut are spending basically our whole careers devoting to that search. We don't think we're wasting our time because things like these meteorites, but that is very different from saying we have proof of life. We only have proof for one example of life on Earth, one. N equals 1 is all we know. Of course, we think that N must be larger than 1, and the meteorites and the organics and the Miller-Urey synthesis and Bob's comments about panspermia and the ability of rocks to carry life from one world to another, even one star to another, are all reasons for hope that N is greater than 2. But if we're going to be scientists, we have to carefully differentiate hope, which may motivate us, and the facts, which is what we have to put in our papers. Bob.
Okay. So I would just add to that one thing. There is one outstanding fact in this whole field, which is this, which is life appeared on Earth virtually as soon as it could. We have fossils going back 3.5 billion years, arguable ones going back 3.8 billion years. So life appears on Earth as soon as it could. And that suggests really only one of two possibilities. Either the processes that lead to the development of life from chemistry are highly probable and it happens as soon as it can, in which case life is very common in the universe, or life is floating around in space and prepare to take over and land as soon as it finds a place that can accept it, in which case life is very common in the universe. So those to me seem, that is the central evidence in the case is life appears as soon as it could, and it either appears indigenously or occurs as immigration. So I think it's very probable that life is extremely common in the universe.
Once again, I want to agree with Bob and actually commend him on a short answer. The way we test between his two choices, rapid genesis and spread, is look at Earth compared to Mars, compared to Enceladus. If we find life on Mars and Enceladus, and it turns out to be the same as life on Earth, that nails it as cosmic interstellar panspermia. If we find life on Mars, life on Enceladus, life on Earth, and they represent 3 distinctly different biochemical systems, that nails it for his other hypothesis, which is that rapid, easy origin of life in suitable environments. So the way to test, the way to choose between models, the way to choose between hypotheses is go get more data. The only place we're going to get that data is our solar system, unfortunately, Mars and Enceladus. So let's go.
And that brings me to my next group of questions, life on Europa and or Enceladus. I did a paper on a project on this many years ago for my culminating project, and all the professors thought I was crazy because it was about life on Europa. But ha ha ha, I'm going to have the last laugh because we're probably going to find it somewhere out there. We have, you know, the Cassini and Galileo missions. They took all kinds of samples and images and spectrometer readings, et cetera, of Europa and Enceladus. So what do the astrobiologists in the audience, on the panel, think about the possibilities, and what have we found that would point to there possibly being life on those moons or any of the other satellites in the outer solar system? Dr. Abramovich.
Okay, I'll take it. I was hoping for Carol to
Okay.
to speak. So from what I've known and from what I've read, basically what we are finding is that NASA had this strategy of follow the water. We did find a lot of molecules of water also on Europa, but also on Enceladus. And what we're looking for in Europa is that we have figured out that underneath 15 kilometers of ice, which is a lot of ice to go through, there's probably a liquid ocean, which is very, very salty. That's the reason why it's liquid, because, you know, Europa is so far away from the sun, its temperatures are super low. And so, and also we are familiar with the environment surrounding Jupiter, which has a lot of a really strong magnetic field. And so we're thinking that these areas, and we're looking at the ice covering Europa, we see that it's formed relatively recent, and it's changing, it's dynamic. So there's a dynamic process going on beneath the ice that then push out new ice to the surface. And hence, once we have this combination of having a liquid state of water, and we have an energetic source in a dynamic place, then we're thinking that maybe perhaps underneath the 15 kilometers of ice, there's enough shelter, there's enough energy for life to evolve in the planet itself. Again, from planet Earth, we know that there are places which are very happy to live in the dark with very little sunlight whatsoever, and they're able to do chemoautotrophs and chemoheterotrophs, and we know that they're very happy using nutrients and other energy sources than light. That's what I know about Europa at the moment. I don't know about any missions going to Europa, which is sad, but I hope it will happen. It takes about 8 years to get there, so it's a really long mission.
I think the Europa Clipper is on the table. They're planning a mission.
Yeah, I don't know if it's going.
Orbiter. That is an orbiter mission. It is not a lander. There's going to be a lander. There was a lander that was pushed by a congressman, actually. And there was quite a bunch of research and study and money was spent on developing that Europa lander. And then that congressman lost his re-election, and that actually put the kibosh on it, which is actually, in my view, a good thing, because it was a very optimistic, success-oriented sort of program, very much in the vein of Viking, that we were going to go to Europa and find life when we hadn't really ever gotten good even imaging of the surface, good enough imaging to know where to land the thing. And Europa is extremely, because of the Jovian magnetic field and the charged particles trapped in the Jovian magnetic field, Europa is an extremely hostile environment. So hostile that this lander was designed to self-explode after a month, so that Europa could not be contaminated by microbes that might be hiding on the spacecraft if the spacecraft were to die because of the high radiation environment it was sitting in. So it was, in my view, it was very unlikely to find life on Europa, because I think it's very unlikely to be there. Certainly on the surface, sitting in this, nuclear explosion level radiation environment. And that it was going to be a complex mission that had a very, very short lifetime. I'm much more optimistic about Enceladus as a life detection target. And Chris would probably be the better person to talk to about this, because he's been the PI of a New Frontiers level mission to go sample plumes of Enceladus. So Chris, why don't you take it away?
Yeah, Carol was a co-I on that mission. It was a concept to fly to Saturn. Enceladus is a small moon of Saturn, and it's really come to the fore in the question in terms of life detection, because Cassini discovered in 2005 a plume of water coming out of the ice in Enceladus, not just water, but organics containing nitrogen compounds, carbon compounds, obvious sources of biological energy. All the requirements, all the major requirements for life are checked, and the samples are coming out into space at a flux approximately equal to the flux of Old Faithful geyser in Yellowstone Park. So all we got to do, so to speak, is fly through that plume, collect the sample. We know we'll get water. We'll know we'll get organics. We know we're going to get material coming from an ocean that's in contact with that hydrothermal system. So Enceladus, to my mind, is the target in the outer solar system for a search for life. And so we are pushing hard on that still. We'll try to put another proposal into New Frontiers to go out there and collect material in that plume and search for evidence of life. In that sense, it is part of the same intellectual exercise that searching for life on Mars is. It's addressing the questions that Bob put forward. Did life start on Earth? Did it come here from elsewhere? To me, that is a compelling scientific question. How do we answer it? We're not going to answer it on Earth. We're going to answer it by comparing life, if we find it on Mars and Enceladus. And as I said before, if those three worlds have life that maps onto a single tree, well, we know that life came here probably in an interstellar comet during the formation of the solar system, like Borisov, the interstellar comet that came through our solar system just a year and a half ago, right? Shedding interstellar dust throughout our solar system. Or if we go to Mars and Europa and Enceladus and Earth and find three different types of life, Earthlings, Martians, and Enceladusans, each with their own distinct biochemistry and genetic code, well, that would tell us that the universe is a wonderful place with all sorts of life forms. Life is easy, it pops up wherever are suitable conditions, and you know, it's a great place to live. We'll see.
Speaking of great places to live, for the astrobiologist on the panel, ice has been found in the permanently shaded craters of the moon and now of Mercury. And I believe the last I read, the ice on Mercury has this organic sheen over the top of it. Is it possible for any microbes to eke out a living in those ice interfaces where it's, you know, on the edge of the ice where the sun is, maybe a little liquidy?
In my view, the answer is a resounding no.
Okay.
So the ice would only be there, certainly on the moon. The ice is only there because it's at 50 degrees Kelvin or something like that. I mean, it's extremely low temperatures and it's sitting in vacuum. I mean, there is no atmosphere. So there is no liquid state possible, even if you could warm it up enough to have it start sublimating. It doesn't go through a liquid stage. So there's no way that anything that we identify as a life form that uses liquid water as its solvent to live in that kind of environment.
Okay, thank you. And I think we might have a little more time. I have several questions from audience members and from a variety of places. I wanted to ask about the perchlorate on Mars. I know that the Viking 1 and 2 labeled release experiment, there's some, you know, we're still going back and forth. Was it life? Was it not? Et cetera. And I know the perchlorate had something to do with that. Can you discuss why that would have affected the life detection experiment?
Perchlorate itself did not impact the life detection experiment. I don't think anyone has actually argued that. Chris, am I wrong about that?
No, you're correct in the terms of the 3 biology experiments.
Yeah.
But it did impact the results of the GCMS.
Okay, the gas chromatograph mass spectrometer.
So what Viking, the contradiction on Mars at the time of Viking and up until really quite recently was that no organic compounds were being found. You know, life doesn't just have its own little the cells, there's all the dead cells. And it builds up over time a organic burden. And without that organic burden being there at all, detectable at all, it was felt by the Viking scientists, most of the Viking scientists, that there couldn't be life there. So that the signals they were seeing had to be some kind of a chemical reaction. And the signals they were seeing behaved more like chemical reactions than biological reactions, in the sense that as soon as you put the nutrients onto the soil, the reaction occurred. Whereas with biology, usually you have to create the exponential growth. So the initial reaction is going to be a few microbes, and then they're going to make more microbes, and so over a course of a few days, you make a big enough colony of microbes that they'll start making a signal that is a strong signal, but you don't see it initially. And on Viking, when you poured the nutrients into the soil, you saw the signal immediately. And then over time, over a few days, the signal kind of leveled off and even died off. So that didn't behave in the way that you would expect microbial growth in soil to behave.
Which would be less immediate.
Right.
Because it would have to metabolize, et cetera.
It would have to grow up more organisms, basically.
Oh, thank you. So what are your thoughts on recurring slope lineae and whether they are salty brines? I know there are some controversy over whether they're salty brines or ice moving or rocks moving down the slopes. What are they salty brines? What's the consensus?
Well, that has been, that story is still evolving. It's kind of been in flux. The latest results show that the recurring slope lineae are apparently thermodynamically triggered. They happen at particular times of year at particular temperatures, but they don't appear to be lubricated flows in the sense of having enough liquid water to actually change the slope that the RSLs would go down. So
we have about 5 minutes.
okay, so basically they behave like dry flows. They only flow to the angle of repose. And if they were lubricated, they would go down shallower slopes. So it really is still kind of a puzzle of what's causing those things.
Okay, so since we have 5 minutes, I want to ask one last question, and then I think we have some Q&A in the queue that I'll get to for our remaining time. What is your for each of you, percentage of possibility extant life on Mars? Dr. McKay.
I would say the chances of extant life on Mars are small, tiny.
Okay.
I'd say 1 in 1,000.
All right. Dr. Zubrin.
I'd say 75%, because there certainly was life on Mars at one time, if from no other source than the Earth, because Mars had liquid water on its surface when there was already life on Earth, and it would have been inseminated from Earth if from nowhere else. And then as the conditions on the surface deteriorated, it would have retreated into the groundwater. So that's my leaning. I think that there probably is life in the groundwater of Mars.
Okay. Dr. Abramovich.
I think that just as I think that the universe is, I'm optimistic, I think it's filled with life, but I think that because it's so far away and because we're not up to it from a technological point of view, as long as we're not digging really deep into Mars and in several places and doing a proper mapping from a biological perspective, I don't think we'll ever find life on Mars at this rate. That's what I'm thinking.
We're not moving fast enough. And Dr. Stoker, what do you think?
Well, I'm closer to Zubrin. I believe that if there was ever life on Mars, there's life on Mars now. Whether it is widespread or not, I think is really the question. And I think it's very likely to take a concerted program to find it. I'm pretty optimistic about the polar ices as a first cut. But if that were to turn out to be a negative result, then I think you are probably stuck with looking into the deep subsurface where there are plausible pockets of liquid water. Now, I have to say that the lake that has been identified under the South Polar layer terrain is not a habitable environment. Just the fact that you have liquid water does not mean you have a habitable environment. And that there is a lot of the scientists who've looked at this don't believe that that's what they are even seeing a liquid water environment, because the temperatures that should be at are way lower than would allow a liquid, even if it were a eutectic solution of perchlorate. So if it's being kept liquid, it's being kept liquid by some kind of a hot spot in that location, or it's some misinterpretation of the data.
Okay.
1 minute.
Oh, go ahead. Oh,
1
1
minute.
minute. Okay, so Daniel Tompkins asked on the feed, do subsurface microbes have a significantly larger biosphere than surface microbes? I guess on Earth. He's
Well,
asking.
most of the earth is underground, so they do.
Okay, all right. I'm just reading the questions. All right, let's see. A lot regarding Enceladus and Europa, the observation from, okay, Arthur C. Clarke, then, okay. Well, we have about 10 seconds to wrap up. Any final thoughts from any of you? I just want to say, first of all, thank you so much for joining this panel. It is a very important work you guys are all doing, and I appreciate it. And I agree, probably 75% chance of extant life on Mars. That's my specialty, and that's what I've been working my whole career to help prove.
I'm happy to find dead life on Mars.
Yes.
Dead or alive, it's fine. In fact, dead is preferred. Very much fewer planetary protection issues. Hooray for being dead.
We're all
Okay.
moving.
Well,
now.
let's go see what's there.
Yes, let's go. Thank you, everybody. Have a wonderful
Thank you
weekend.
very much.
Thank you, Nicole.
Thank you. Amazing work.
Thanks.
Thanks.
Thank you.
Okay,
Ciao.
bye.
Bye.